He was there for a few years. Then he was recruited to Yale in 1991, where he is now the Ellison Richard Professor of Ecology and Evolutionary Biology. So Günter is prolific. He has written more than 400 papers and 4 books. His main research interests are in the areas of evolution of development, evolvability, evolution of complex traits. His most recent book, published in 2014 by Princeton University Press, was titled Homology, Genes, and Evolutionary Innovation. And this is really on the fundamental concept of homology that all evolutionary biologists care about. And this book won him a prize. I have to look at the notes. So the prize was called Daniel Elliott Medal from National Academy of Sciences. So this medal is for the best book in zoology or paleontology in the past 3 to 5 years. So that's a great honor. And Günter, because of his prolific work, original work, he has won many honors and prizes. He was a MacArthur Fellow in 1992, and he was a member of the National Academy in 2018. I met Günter first in a conference exactly 10 years ago in Stony Brook, where Dr. Futuyma and others organized a celebration of the 150th anniversary of the publication of Darwin's book. And in that meeting, we found that we share a common interest on pleiotropy. So we wrote a review paper for Nature Reviews Genetics 2 years later on the topic of pleiotropy, which turned out to be very controversial. Some people criticized us. They wrote letters to Nature Reviews Genetics. I've never heard people criticize a review paper, but they did. But anyway, our review paper was very successful in the sense that it attracted interest, and it's also very well cited, right? So, so today Günter is going to talk to us.
We don't say whether it's positive or negative side.
Yeah. Okay, I don't see the title here, but yeah, it's something
I can do that.
Pregnancy.
Here.
So it's the evolution of mammalian pregnancy, the path from pathology to physiology. Please welcome Günter Wagner.
Thank you very much. Thank you very much for the introduction and the invitation. I had a really great time talking to people here. It's a really, impressive and vibrant intellectual community here, and I'm really glad to have the opportunity to interact with many of you. It's a really great, opportunity. To be here. It's not my first time I'm here. I forgot when I was here. Miriam, to remember, it was 20, 30 years ago or something like this. Yeah, right. Okay. Okay, so today I want to give you an overview of our work on mammalian pregnancy, and I will not introduce how we came up with working on pregnancy, because I explained it to some students during the day, but actually emphasize a view of how a complex trait is evolving from a probably coincidental pathological reaction rewired into a physiological pathway. So if you think about pregnancy and viviparity, in particular human one, it all seems so obviously straightforward because you have a specialized structure, the placenta, that mediates the growth, the transfer of nutrients, hormones, oxygen to the fetus, and then also the removal of waste products from the fetus to the mother, then doing the things. So that seems to be like a good idea, right? So it should actually. One asks yourself, why doesn't every animal is doing the same thing? And this notion that viviparity is a good idea is also supported by the fact that viviparity is evolved many times. So it seems to be a good, of course, in lizards like Lacerta vivipara, at least back then it was called Lacerta vivipara, which the name already tells you what reproductive mode it has. You even find it in sharks, in lots of skinks and so on. So, you know, it's the exact number how often it evolved, even in vertebrates, is still controversial because it depends very much on details of the phylogenetic tree that is still in flux. But among all of the cases of viviparity, the mammalian, or let's say eutherian pregnancy is special. And here mammalian is under quotation marks because what I actually mean is eutherians. When we talk about, you know, mammalian reproduction, it's usually eutherians, and other mammals actually do different things. And one of the things that is special, and to my knowledge, it's only found in eutherians. And if you know any examples outside of eutherians, let me know, because I haven't found any examples outside of it, where the development of the fetal-maternal relationship takes place under destruction of part of the maternal tissue. And that is something that, you know, is quite special, and it is special for the following reasons. So if you look at what is it? Lizards. This is not working. In lizards, so here's the histological, here's the maternal side, here's the trophoblast, and the relationship is very superficial. It's just an attachment, not even an attachment. It's an apposition of the fetal membranes of the fetus to the uterine tissue, and the fetus is actually kept in place by muscle tension rather than being actually anchored to the wall of the uterus. And here you see it's also quite transparent. This is the skink fetus in the uterus. You can actually see through it. Okay. So I don't know why I have this here. I forgot about this. So actually, the main point that I wanted to make here is that, you know, if you think about traits that involves destroying existing tissue of the mother, the generic expectation is that by compromising the tissue integrity of the mother, you would expect that the mother has an inflammatory reaction to compensate for the destruction of maternal tissue, and that it activates neutrophils and would actually destroy anything that sort of tries to invade into the maternal tissue, right? And that in eutherians obviously is not taking the case. So there is a physiological barrier that needs to be overcome in order to be able to have a sustained fetal-maternal relationship that actually starts with destroying part of the maternal tissue. So here is an overview of what I'm going to talk about. First, in order to situate the situation in eutherians, I found it helpful to actually look at the closest relative to a sister taxon that has a form of viviparity but doesn't have invasive placentation, right? And then ask the question, we'll spend some time on this, and then ask in what way are the eutherians different from what we learned in the opossum, South American opossum, Monodelphis domestica. And then I propose a scenario of how that eutherian situation could have evolved, and this is from another lecture, forget about it. So let's talk about pregnancy in the opossum Monodelphis domestica. Of course, you know, one reason to go there, just to remind here, this was from a talk that I gave to MDs, so I had to bring more comparative biology in there. But anyway, just as a reminder, mammals have these three major groups: monotremes, egg-laying marsupials, viviparous, but special in a way, and then the placentals, where, you know, all hell breaks loose and you have this extended gestation and invasion and things like that. Of course, the outgroup that we need in order to understand what's happening here, what's probably happened in this part of the tree, is the marsupials, and within the marsupials, of course, there are various groups, quite a number of them, Lucky to us, the most basally branching lineage, and which is also in its reproductive biology probably quite ancestral in many respects, are the opossums. And while in the macropod it's the wallabies and kangaroos that are studied a lot in Australia, they are actually highly derived. They have an extended gestation, recognition of pregnancy, monoovular, the only ovulate one, while down here in all the other groups are polyovular, short gestation, no physiological recognition of pregnancy. So lucky for us in North America, actually the only marsupial that we can have in the lab reliably is a relatively primitive marsupial with respect to female reproduction, within limits, of course. Okay, so here is an overview. So let's say copulation is our day 0, then fertilization is happening a little bit later, and then gestation is about 14 days, and the 14 days lead to the birth of highly immature neonates. It's about the size of a very small jelly bean, and it's very immature because they don't even have hind limb buds. They only have forelimbs in order to crawl up, no hind limb buds. It's very, very immature. The placentation is epithelial-chorial, so you have the uterine epithelium, the placenta, or the trophoblast, and they can easily be detached from each other, so it's non-invasive placentation in the sense that there is no sustained invasive phenotype. You will see later why I'm making these qualifications. And here it's just showing how small they are, and then they don't have hind limbs, and this is how they look. It's a marsupial, but doesn't have a marsupium, so you can actually count the neonates attached to the things. Now, that's of course in major contrast to what Eutherian means. Many of them can be actually quite mature when they are born. It's also the case that if you look at hormone profiles, you cannot distinguish by hormone profile and female that is in its estrus cycle versus the one that is pregnant, which is rather inconvenient to us because we usually don't know whether a female is just in pseudo-pregnancy during estrus or actually pregnant. But it's also one of these primitive characteristics. Another characteristic is that in Monodelphis, the eggshell coat, technically spoken, persists for about 12 days of a 14-day pregnancy. So here you have the uterine epithelium, here is the trophoblast, here you see the shell coats physically separating the fetal tissue from the maternal tissue at a day 11.5 post-copulation pregnancy. And then the next day they start to falling apart, and here's a few other things that at the end of day 12 the trophoblast attaches to the uterine epithelium, and then a day later it starts to separate again, and then on day 14 you have parturition, you have birth, right? So even though it is a 14-day gestation, the actual physical interaction between maternal tissue and fetal tissue is only for 2 days, right? For most of the time, they are separated by the shell coat. They may communicate chemically, but it is not a direct physical interaction like it is in Eutherians. So we have 14-day gestation, intrauterine hatching and attachment to the uterus at 12.5 and birth around day 14. So that is quite different from even a mouse gestation where you have 21 days or 19 days depending on the strain, but attachment and implantation happens at day 4, and most of the time they actually are in close connection between the fetus and the mother. It's interesting that there's a lot of changes actually happening in pregnancy and in estrus cycle that are quite similar in terms of histological changes. There are only two. The only two differences that we could find is that in pregnancy, the uterine glands became active, while histologically in the estrus cycle, even though the uterus is increasing in size by factor 2 or whatever, the uterine glands remain dormant, and in pregnancy, there's this subepithelial stroma gets replaced by capillaries, by blood capillaries. But otherwise, there is a lot of automatically happening changes in the uterus, even though there are no embryos around. So we also looked at this from a transcriptomic point of view. So in estrus cycle, non-pregnant and early estrus cycle that's corresponding to mid-gestation is barely discriminatable. But in late gestation, it actually changes a lot, and this is the trajectory in pregnancy. And there are great similarities, but a few differences. And actually, we were interested in exactly finding out what's different between late-stage estrus and late-stage gestation. So what is contributed to the thing? So some of the things, so this is sort of a way of representing GO analysis, gene ontology analysis. There's lots of upregulation of things that make a lot of sense because, you know, if the fetus is there, you want to transfer nutrients, so triglyceride metabolism, fluid level, and ion transport are upregulated. So that's sort of expected given the function of that. But what, and sort of they are actually not, so this is based on RNA, you know, you find some of them, you know, the fatty acid binding protein is expressed in the protein, is in the glands, and the aquaporin in the luminal epithelium, and so that all makes sense. But what is more interesting is that at this time when the fetus starts to attach, you have also a massive upregulation of genes related to the acute inflammatory response, right? So if you look at it in more detail, we have whatever 250 genes upregulated related to immune response, acute inflammatory response, and so on. And here you see the various clusters of genes in sort of a semantic space that shows you, you know, how they are related. And here you have examples of those with pro-inflammatory cytokines, IL-1 alpha, IL-17 alpha, IL-17 tumor necrosis factor, also some anti-inflammatory cytokines, IL-10. So you see here the RNA levels as they go up from mid-gestation to late gestation. Interesting also is COX-2, which is the enzyme, the critical enzyme for producing prostaglandins, which are of course also inflammatory mediators. The protein is expressed in the luminal epithelium. Here's the RNA expression of it. And we also actually find prostaglandin E2 in the uterus, in the pregnant uterus. So this is non-pregnant and so on. And that's also a similarity to a situation that we find in human implantation and also in the mouse, where also the epithelium during the implantation phase expresses this COX-2 enzyme producing prostaglandin E2. Now, prostaglandin E2 is not produced in one step. It's one that you get from arachidonic acid. You get an intermediate, this PGH2, and then you need another enzyme to make actually the PGE2. Now, surprisingly, actually, if we look for the localization of PGE2, this last enzyme in the cascade, we actually find it in the trophoblast. And that is something I will come back to later. So the question actually, it's actually a potential way of how the uterus can know that there's a trophoblast or a fetus there, because the uterus seems to produce PGH2 with COX2, but the last conversion of this here into the active prostaglandin seems to happen in the trophoblast. So only if you have a trophoblast there, you get PGE2. That's sort of a way of sort of local recognition of pregnancy. That is somewhat controversial because people say that's not possible, but that you have the PGH2 go from one cell to the other. But I think it's still an open question because there are transporters that can move PGE2 around from one cell to the next. And actually, if you look at the expression of this transporter, it is actually expressed in the luminal epithelium. This is a in situ hybridization on the frozen section of the uterus. So what does PGE2, what role does it play? In general, PGE2 is known for causing edema because it causes vascular leakage, which may enhance nutrient transfer to the uterus. At least that was my original hypothesis, but it turns out that we couldn't support that. So if we inhibit the production of PGE2 by inhibiting COX2, it actually does not affect the survival of the fetuses. So we actually don't know whether it has a functional role in opossum. Another one that is interesting in this context is the expression of interleukin 17A is also started to be expressed after the attachment of the fetus, and interleukin 17A is interesting because it is the main pro-inflammatory signal in mucosal surfaces. For instance, if you have an inflammation in the gut, then through various stages, it leads to the recruitment of neutrophils, and the neutrophils are sort of these marines of the innate immune system. They come in and shoot everything to death. And it's also sort of problematic because they cause a lot of, they can cause a lot of collateral damage because they are so indiscriminately destructive. Actually, we do find cells that look like neutrophils in day 14 uterus of the opossum, and that is consistent with that finding. So summarize what we have found in opossum is we have this 14-day gestation, a long pre-attachment phase of about 12 days, then attachment with the activation of inflammatory mediators, then leads to the recruitment of neutrophils, and then parturition. So one interpretation of this is that in the case of opossum, the attachment and inflammation phase is actually a precursor to and contributing to parturition, because even in humans, parturition, so giving birth, is an inflammatory process where macrophages and neutrophils actually invade the uterus and then lead to the cramps that then lead to the expulsion of the neonate. So in a way, don't quote me on that, the parturition can be seen as a similar mechanism as diarrhea, right? You have a pro-inflammatory environment that leads to activation of inflammatory processes that lead then eventually to smooth muscle contraction, and then the expulsion of whatever is causing that irritation. And you could make an argument that there is a similarity between diarrhea or vomiting and giving birth from a cell biological point of view. Okay. So the next question, of course, is why would the maternal tissue react to the attachment of the fetus with endometrial inflammation? And of course, what you need to realize, mostly we study inflammation as a part of the response to infections. But as we all know, infection is not the only way how inflammation can be activated. Tissue injury without infection or tissue stress can also activate the same pathways. So if you have you sprain your ankle when you go running, you get a tissue injury inside your ankle. There is no infection there, but you still have swelling and all the inflammatory processes going on. And also tissue stress, like in obesity, leads to tissue stress, that leads to inflammation, leads to diabetes, right? So inflammation is probably only secondarily related to infection, but primarily a way of reacting to compromises of tissue integrity, right? And if we think about that way and realize that both Eutherian as well as Opossum and Marsupial early embryos actually have to hatch out of an extracellular coat like the zona pellucida or the shell coat in marsupials, and the way they do it is by secreting proteases, okay? And so here, the interesting thing that these proteases, they continue to be expressed after they attach to the uterus. So here you have an immunostaining for a particular serine protease. This is the name. The red staining here is for this protein here. This is the trophoblast as it is attached to and close to the uterine epithelium. So the embryo produces enzymes to destroy the shell coat, but continues expressing and probably secreting these enzymes after they have attached to the mother. And my best guess, although I haven't tested it experimentally, is that the inflammatory reaction is actually a reaction to this, continued secretion of tissue-destructive enzymes. Now, this property of expressing these enzymes is one that is conserved all the way up to human. This is data from Jan Brosens in the UK, where green is a immunostaining for a similar protease that is expressed specifically in the trophoblast of the early embryo. And there is also some evidence from histological evidence that I have also found in our sections, is that shortly before birth, the trophoblast of the opossum embryo actually starts to breach the epithelial barrier to the stroma, as you can see here. And I also saw it. I just didn't find this microscopic slides where I found it, so I used this here, which shows that even though technically it's not invasive, the trophoblast still has the ability to compromise and attack the maternal tissue. Now, the main difference, of course, is that this event happens immediately before birth in the opossum, while in the humans, this invasion is actually one that is maintained for whatever 9 months or whatever it is. So the idea is that attachment is associated with inflammation, probably because it leads to tissue injury, and tissue injury in the endometrium generically activates inflammation, and inflammation can have the effect of leading to smooth muscle contraction and then to birth. So this is the situation, summarizing with this one more line here, that probably this inflammation is probably caused by fetal proteases. And so far, I have told you a story where the reaction to the presence of the fetus is entirely sort of a passive one on the side of the mother. But actually, as it's already pointed out, that part of the inflammatory reaction, namely the production of prostaglandin E2, actually seems to be cooperative between the mother and the fetus, where one enzyme is produced by the mother, the COX-2, and the other, where is it, PGE2 here, and the other enzyme is produced by the trophoblast. They actually cooperate in enhancing this inflammatory reaction. And actually then it became to an unavoidable conclusion, because I told you about this highly inflammatory IL-17 interleukin, which is going up. But when we looked, so Daniel Stadtmauer, a graduate student in my lab, looked in where this is expressed in the pregnant opossum, it's actually produced by the trophoblast. So the trophoblast itself is also contributing pro-inflammatory signals, probably because that's the way how it initiates the parturition at the end of pregnancy. So what I think in the opossum is happening, what we're seeing is a cooperative inflammation where cells from the fetus and the mother are cooperating in producing an inflammatory state that then leads to parturition. For instance, the COX-2 in the luminal epithelium, PGGES in the trophoblast, shuttling back prostaglandin E2, probably so IL-17 here, maybe caused by the expression of IL-1 and IL-6, but that is hypothetical, but then leads to neutrophil recruitment and then to this full-blown inflammatory process. Okay. So here's the phylogenetic, first part of the phylogenetic symposium model, and that is, I think, what happened in the hypothetical Therian ancestor was a generic information in reaction to the presence of these proteases that the fetus brings, and then there is a split into the marsupials, where this generic inflammation becomes sort of part of the parturition physiology of the, and to make sure that it is working well at the time when the fetus needs to be extruded. There's this cooperative inflammation where both fetal as well as maternal tissues interact with together to lead to this inflammatory state that then ends in parturition. So the question is, what is happening in mammals? So obviously this is a statement that what's happening here is homologous to a generic inflammation, but one that has been evolutionarily modified to make sure that parturition is happening. What is the difference between opossum and placental mammals or eutherians? Just a little bit of a reminder. I think most of you know that in eutherians we have different degrees of invasiveness. Non-invasive where just the trophoectoderm attaches to the uterine epithelium, and the most invasive one is the hemochorial form, where both epithelium and mesenchymal and blood vessels get destroyed, and the maternal blood is in direct contact with the trophoectoderm or the placental of the fetus. If we do a phylogenetic reconstruction, we come to the conclusion that the most recent common ancestor of all eutherians probably already had a hemochorial placenta, and that's that the non-invasive placentation that we find, for instance, in cows and so on, is actually secondarily evolved, but that's a different story. So these animals that have less invasive placentation are actually not primitive as people thought for a long time, but they're actually secondarily non-invasive, and that's for another seminar. And gynecologists and obstetricians have known for a long time that some part of inflammatory reactions is actually a necessary part even of human pregnancy. That is, my former colleague Gilmore, for instance, has pointed out that the beginning of the pregnancy is an inflammatory process, then you switch to an anti-inflammatory growth phase, and then, as I already mentioned, parturition itself towards the end of pregnancy is one where inflammation starts again and eventually leads to the extrusion of the neonate. And that is rather puzzling to the community, because at the same time as in certain phases of human gestation, inflammatory type processes are necessary, they are also one of the biggest enemies for the continuation of pregnancy. One of the major ways of how prematurity to early birth or pregnancy loss is happening is through various ways activating inflammatory processes if they happen at the wrong time in gestation. And we actually also looked at our idea then was that probably what's happening in humans and in other eutherians is actually another form of the evolutionary modification of the generic inflammatory process that stands at the beginning of the evolution of pregnancy. And we compared, for instance, transcriptomes from our opossum things with, where is it? This is, I think, don't know what animals this were. A human, up in human, 3 different microarray studies. They have detected fewer genes because microarrays are not as sensitive, but there is quite substantial overlap in the activation of genes that we find activated in the opossum and the ones that get activated in humans during implantation. So that sort of supports the notion that these two processes are not identical, but they are probably homologous. So our model for the evolution of eutherian pregnancy is that there's something similar to what we see in the opossum, probably in the common ancestor of marsupials and eutherians, and that the key innovation of the eutherian pregnancy is the intercalation of an anti-inflammatory phase of pregnancy between the attachment and inflammation phase during implantation and the late secondary inflammation that happens and then leads to parturition. So the key innovation was to allow inflammation early on in order to allow attachment and implantation of the fetus, and then switch off the inflammatory process, suppress inflammation in the mid phase in order to enable a sustained pregnancy that's much longer than in the marsupials. And that's only possible by avoiding inflammatory processes and exclude neutrophil extension. And then, in order to end pregnancy, all what you need to do is to remove the anti-inflammatory support for the maintenance of the fetal-maternal interface. Okay, so that's our model. That what's actually happening here is this is the key innovation. There was inflammation at the beginning. Key innovation is the ability to turn off inflammation for long enough so that the fetus can grow to the size that it needs to. Then remove the support, and then you get parturition, in probably in the same way as you have it in the opossum. So what we need to do in order to test this a little bit is so most of the model organisms for reproductive biology are in this clade. We need to get here somewhere in deeper clades, Afrotheria and Xenarthra, and that is not a particularly attractive proposal, because the choice of animals down there is the elephant, the dugong, the sloth, anteaters, and so on. Not very attractive for an experimental biologist. But again, we are lucky living in the US. We actually do have an animal that belongs into this group, and this is the armadillo, which is a Xenarthran, and phylogenetically belongs here. This is an older phylogeny, probably they are sister groups, so they are actually belonging to the most ancient eutherian diverging lineage. So we have, those are the things, that is, whatever. So non-pregnant, mid-gestation, and early gestation or implantation. So there's some diapause here. So we have samples for all of those. Of course, that requires for me as an experimental biologist to get out of my comfort zone. So I had to become blending in with the Texas local community with my own gun and my camouflage in order to harvest opossums and then, produce, get tissue samples to investigate. So interestingly, you remember this expression of IL-17. We do not find it at any stage of the gestation in opossum, even though other inflammatory mediators are expressed in various stages of development, but the key difference is the basic absence of this IL-17A, which is the key mediator of mucosal inflammatory responses. And that corresponds pretty well with the presence of neutrophils in the uterus during pregnancy. Neutrophils we found in the opossum and IL-17A, even though it is produced by the fetus, but not in any of the eutherians here. We found evidence for a similar recruitment of neutrophils early to mid gestation. So one hypothesis that we derived from that is that a key innovation could be an evolved suppression of IL-17 production during implantation, which would be normally part of the mucosal inflammatory response linked to neutrophil recruitment. So how would this have evolved? And we think it has something to do with another innovation, namely a new cell type called the decidual cell. So these cells are the decidual cells in I think is a human endometrium. And there are endometrial stromal fibroblasts in the marsupial, but these cannot differentiate into the decidual cells. And we know that decidual cells are actually necessary for implantation and the maintenance of pregnancy. So Arun Chavan looked at this idea, so that tissue injury activates macrophages. They secrete interleukin-16, which then inhibit the differentiation of a naive T cell into regulatory T cells, which are anti-inflammatory, but enhances the differentiation of naive T cells to Th17 cells, which are then inflammatory. So the invention of the role of the decidual cells could be either inhibiting the activation of the macrophages or inhibiting the differentiation of the T cells into Th17s. He already produced data that shows that this is not the case, so decidual cell conditioned media don't influence macrophage activation. So that's not the question. So the question is whether that is correct. And so he did the following type of experiment, taking naive T cells, those are human cells, differentiate them into Th17s, either in control media using these signals here, or in the presence of conditioned media from human decidual cells that we mix into this. And here are the results. So this is the control experiment without activation of the differentiation into Th17s. If you do differentiation medium, you get an increase in IL-17 secretion. If you add 50% control unconditioned decidual cell media, it doesn't change much, but if you have conditioned media, you go down to this. So this suggests that decidual cells have, one of the roles of decidual cells is actually to suppress the mucosal immune response, the part of the immune response that leads to the recruitment of neutrophils. Another sort of indirect evidence is that usually mucosal tissues have what's called mucosa-associated tissue or MALT. This is sort of like a lymph node within the mucosal tissue itself, but in the endometrium, it's extremely rare. You only find it in maybe 1%, 1 per mil of the cases. This is evidence from a colleague of mine from the gynecology. So under certain conditions, in particular postmenopausal women, there can be MALTs developing, but a healthy woman in reproductive age usually does not even have the tendency of producing this mucosa-associated lymphatic tissue. This is sort of so the question is, do the residuals suppress the formation of melts and therefore the activation of T cells there? Another evidence from other people. I don't want to go through this. I'm thinking I'm overdoing my welcome here, but our conclusions that the residual cells of one of the first roles that they evolved for was suppressing the inflammation in the endometrium after the initial implantation of the embryo. So here is a scenario. So we have this part of the scenario already discussed, from the hypothetical generic inflammation caused by the fetus to the cooperative inflammation that we see in the opossum, which does not have the anti-inflammatory phase that allows it to extend pregnancy, to a case where anti-inflammatory phase is increased by a maternal suppression of inflammation. And in order to sort of contextualize this, it's important to understand that of the total inflammatory cascade, only parts of it are actually deleterious. Angiogenesis, vascular leakage, permeability are actually good for the embryo. Also, recruitment of some forms of regulatory immune systems, and so on. So the main evolutionary thing was that to have inflammation, maintain the parts of the inflammatory process that are actually positive for the fetus, but suppress the recruitment of neutrophils, because those are the destructive things that would attack the placenta, and also, at least for a while, suppress the production of PGF2, so another prostaglandin that has the property of causing smooth muscle contraction, so that you can understand the implantation process as a modified generic inflammation which suppresses neutrophil recruitment and suppresses for a while the myometrial or the smooth muscle contractions. But these parts are probably maintained, and therefore we still can recognize, even in human pregnancies, an inflammatory signature in terms of the signals that are exchanged. But of course, it's not a full-blown inflammation, because otherwise the fetus wouldn't be able to survive. So that leads us to this scenario that we call the inflammation paradox, where the primary obstacle for the evolution of implantation is inflammation due to the compromised tissue integrity caused by the embryo. Implantation evolved by the endometrial reprogramming of the mucosal inflammatory response, and also as a side effect sort of explains why human pregnancy is so vulnerable to compromises of suppressing the inflammation, because inflammation is at the beginning of the process. It's always sort of in the background. It's only suppressed to the degree to allow extension of pregnancy. But if this suppression gets compromised by infection or whatever, it's catastrophic for the continuation of pregnancy. So the consequences of these models is the primary responsibility for tolerating a fetus at all comes to evolutionary changes in the endometrium and the decidua, and placental evolution and immune camouflage, that is sort of the main focus of the reproductive immunology after Medawar, is actually a secondary consequence, or at least both physiologically and evolutionary secondary, that only became relevant after gestation could be extended long enough so that adaptive immune response can actually kick in. And that is actually so in Opossum, adaptive immune response takes 19 days. It can never happen during the phase of the short gestation. So if that was too much detail and confusing, I have a cartoon summary for you to sort of take home here in 3 slides. So at the beginning was inflammation. Then the mother tamed inflammation. Hestia is the Greek goddess of fire that tames that contains it so that it can do its work in the kitchen. And then the placenta takes all the credit because it is sort of the more obvious organ out there and actually gets credited with the evolution of pregnancy way too much, because in my view the placenta could only evolve after the maternal genome evolved to be able to actually tolerate an invasive organ in the endometrium. And with that, thank you for your attention. This is some of the players in this endeavor and the funding sources for some parts of this work. And thank you for your patience and attention. Please.
As a boxer, not knowing anything about animals, I'm just curious. What is the evolutionary advantage of live birth?
Live birth. Yeah. So if it just take live birth as a broad category, not so much about the particular form of placentation that I talked about, and if it take evidence from reptiles, from squamates, from lizards, then it looks like viviparity is favored under conditions where the environment gets cold. So either in higher latitudes or altitudes, there you have a higher frequency of reptiles because you can carry around, and then you even have some metabolic warmth to incubate them. So that is the main ecological correlate that I know of. There might be others, but this is the one that has been documented in lizards.
Yeah, that makes sense.
Okay. The first thing that makes sense in the seminar.
The seasonality, appears in seasonality.
Right, as a way of sort of as sort of avoiding. And it's also interesting. There are polymorphic populations where the same species has life-bearing and oviparous populations in Yugoslavia, former Yugoslavia, and the karst, and that they are also high up in the mountains and low down. So it could be that they actually adaptively change their reproductive mode depending on the environment. Yes?
Thank you for an interesting talk. I'm just wondering how representative the possums are for the variety of different types of placental organization you get in marsupials. Is there additional variation? And sometimes it's hard to identify specific traits as plesiomorphic or
I admit that I'm not fulfilling the cladistic criteria for a primitive trait state, but I think there are ancillary arguments that support that is as good a representation of the ancestral condition as we can. So there is no evidence in mammals that where they evolved back to viviparity or shortened So once you have it, you're fixed with that. The other thing is that the whole suite of reproductive traits, as I made the point in opossums, is primitive within marsupials, and the only exception are the macropodids, which have evolved extended gestation up to 23 days. Not very much, but still more than the cycle, and also are monoovular and have a hormonal recognition of pregnancy, which the opossums, paramelids, and the others don't. So they are certainly, in many respects, primitive with marsupials. It's about as close to the Therian ancestor that we can get with any extant animal. So I mean, I would love to have more intermediate stages and other, but there are only 2 sister taxa that you know we hopefully can use you know functional criteria to sort of support the notion, even though we don't have the strict cladistic criteria for primitiveness.
It seems to me that the origin of the placenta should come at a relatively high resource cost for the mother. Do we see any sort of impact on, say, the number of young that the mother can have when the placenta originates from an evolutionary standpoint? Do we see any sort of ramifications of that cost?
I mean, it's a shift of the resource allocation, right? Because in oviparous species, you invest into the egg, right? They are much larger, have yolk. Eutherian or even therian eggs don't have any yolk at all. They don't even have yolk proteins in the genome. So I don't think that the overall investment is different. It's sort of, you could even argue that in the case of extended pregnancy, you can sort of cash out your contribution over a longer period of time rather than putting everything into the oogenesis phase. I wouldn't expect that the number of young is much constrained by because mice have whatever 12 to 20 offspring, right? Pigs have how many they have, 18 or whatever. So they are able to produce a lot of neonates even with this form of nutrition. Yep, sure. Yes, please.
Gestation varies across mammalian diversity, the length of gestation.
Right.
And so I'm curious about the sort of the end of this modified inflammatory response and the trigger for it. And
In the eutherians, you mean?
In the eutherians, and you suggest stresses, but surely there must be some sort of genetic programming as well.
Right. I mean, that is actually one of the big scandals of reproductive biology, that we still don't have a good answer on this. And that's a scandal because one major public health crisis that we have is a crisis of prematurity. And in order to prevent that, we would be good to know how that's regulated. But that turns out to be not the case. We have not a very, but one way of thinking about it is that the parturition gets triggered in at least in humans when the fetus gets into stress because the mother cannot supply enough energy to maintain the large fetus. And then that leads to the activation of the pituitary adrenal axis that then activates the production of surfactants, and the surfactants get into the amniotic fluid, and that activates the endometrium over steps, and then you get the withdrawal of the anti-inflammatory support through functional progesterone withdrawal. I mean, with humans it's particularly even worse because in most mammals you have, so in eutherians, but not in marsupials. In eutherians you need progesterone in order to continue pregnancy. And in many animals, towards the end of gestation, progesterone goes down, for one or the other reason, one of which is that if the corpus luteum degenerates, and then you have a natural way of getting to it, or maybe even but in humans, the progesterone is produced by the placenta after an initial phase, a transition from luteal placental transition in the production of progesterone, and actually the progesterone levels in the blood is going up all the way to parturition. It only goes down after birth is delivered, and the organ that produces progesterone is. So in humans and in other higher primates, we have even a bigger problem to solve, namely how do we get parturition with at very high levels of systemic progesterone in the blood. And so those mechanisms are probably evolutionarily much modified compared to mouse and other eutherians, because they have an additional, they make pregnancy more vulnerable to stimuli, because they just withdraw gradually the support that is given to gestation by the presence of progesterone. So unfortunately, there is no good answer to that, and it's actually a big scandal, because it also means that we don't know exactly how to help women that are in danger of prematurity of the babies, how we can support them to avoid premature birth. So it's still rather murky of how that works. I mean, there are many details that are known that you don't want to hear right now, I'm sure, but it doesn't make a whole lot of sort of global sense as an explanation. Yes.
So this may be pushing it too far back, but how does the monitoring know when you're laying eggs?
Yeah, that's a good question. And actually, there's a nut. So you're very good at identifying the big holes here, and that is, we should actually make we would need to understand the evolution of parturition also as a consequence or as a modification of oviparity. So how and when eggs are laid, right? And it turns out that even in reptiles and in birds, which of course are also reptiles, the PGF2 is a major way of how to, the uterus gets contracted and to push out the egg. So the lazy answer is that we know almost nothing about monotremes, right? Because they are so protected in Australia and New Zealand, and there's basically, as far as I know, no published work that actually investigates the molecular biology of their reproduction. The only things that we have are fairly old monographs from the 70s that are mostly natural history, which is important, and morphology, but very little mechanistic work. Yeah.
I don't like to hear the monograph from the 70s.
It's not old to me, but but for the rest of the audience. But what's old in the sense that it's not reflecting a lot of genomic and molecular biology. Nothing, basically. I know that there's one group in Melbourne who is starting a project on, I think, echidnas, but that's just a starting project. It is too little known to be discussed.
Okay, with that, I would like to thank Günter again for an interesting seminar.
Thank you. Thanks for
So that, do you think that's the major switch? Is it possible to test it in animal, like you see off, see what would happen? Like would it affect the gestation time? Length?